All right, guys, welcome back. It's gonna be kind of weird doing this like this, but we'll give it a shot. So we'll start off going through this week's quiz, which hopefully you did yesterday. And yeah, so complete the diagrams. The central what of molecular biology is what we started talking about last week, and we'll be talking about this week. Um, the answer to that is this essential dogma. And a dogma is a firm held belief, something you believe to be true. Now, the next step was to fill in these steps of the central dogma. You have your diagram of a molecule, and you have your nucleus as this inner ring here, and then you have your cell wall, outer ring here. And so, step one starts off as what? Well, it starts off as DNA. That's where the information is. And then from DNA in the nucleus, through the process we talked about last week, it gets copied or it gets copied to mRNA, to the messenger mRNA. So that copy then leaves the nucleus, goes out into the cytoplasm, and it creates a protein. And that's the process called translation, which we'll be talking about today. But the process of copying from DNA to mRNA is called transcription. Alrighty. Now, we also talked about a bunch of locations and items in that process. And so we'll fill out the diagram on the right here. We have our little nuclear membrane. That separates the nucleus from the cytoplasm. We have a little port in there that allows the mRNA to escape. So that's our nuclear pore. And this is inside of the nucleus. This little strand we have here that's being worked on, that's our mRNA. And then we have out here is a cytoplasm. And we have these two pieces of something that's clamping down on the mRNA. And so they both form a ribosome. But we have two different parts of it. We have the large ribosomal subunit, and we have the smaller ribosomal subunit. And so that's that diagram. Now, ribosomes are made out of what? Um, are they made out of mRNA and simple sugars? No. Um, are they made out of mRNA and proteins? No, not quite. Are they made out of rRNA and proteins? Yes. So a hint there is you have that R at the beginning of your rRNA, which matches with the R in ribosome. And it's not pure RNA. Ribosomes, they also have proteins, which help provide structure and other things for it. A polypeptide is a chain of what? Is it a chain of amino acids, codons, nucleotides, or ribosomes? Well, we know it's not a chain of codons. If we have a bunch of those are those form our genes. We know nucleotides are not; those are our monomers. Those are pretty simple. That's a base, a sugar, and a phosphate. Um, is it ribosomes? Um, no, there was a different word, a polysome, which described a chain of ribosomes on an mRNA. So it's not that one, and that will leave amino acids. So a polypeptide is a chain of amino acids. Now, an intron, does an intron get kept or does an intron get discarded? That's a good question. Well, as it so happens, an intron is yeeted. It gets gone. And so if an intron is discarded, then an exon is kept. So there's our quiz for this week. Make sure you bring those in for next week, just so we can keep tabs and see how everyone did. But if you got all the diagrams and stuff correctly, that'll be a big portion of it. So good job. And yeah, let's move on to this week's content. So we're going to be talking about chapter 17. And this is our next process in our central dogma, translation. So we have our central dogma, do. We have DNA going to mRNA through the process of transcription. And we have our mRNA being translated into a protein in the cytoplasm. And today we're going to talk about that process, how we get from mRNA to a protein. Yeah. So here's how it works. We talked about ribosomes a little last week and how they build proteins from our mRNA, and now we're gonna look at how that actually happens. So your mRNA comes out of the nucleus, and it's just a little strand of mRNA that's floating out in your cytoplasm. And what happens is a bunch of ribosomes start attaching. Boop, 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 boop. And as they start moving down the chain, so they'll start one end and then they'll go to the other end. What happens, you're going to have it's going to keep growing. This is your polypeptide chain. It starts small, and then as the ribosome moves, it gets bigger and bigger as it collects and decrypts and translates all these little codons you have on your mRNA into amino acids and puts them together to form our polypeptide chain. Term that we're given here is a polysome. So a polysome is when you have multiple ribosomes attached to an mRNA strand. Alrighty, so that's our definition of a polysome. Now, so far we've covered a couple different functions of RNA. We know that RNA is used to make mRNA, which is a messenger. It brings information from inside the nucleus to outside the nucleus. We learned last week that we have rRNA. Now, rRNA makes up ribosomes. So there's two, and now we have another one. This one is called tRNA. Any guesses what the T is for? Well, tRNA is transfer RNA. And so what tRNA does is tRNA is what actually brings your different polypeptides, your different amino acids, to your ribosome in order for them to build the proper polypeptide chain based on your codons. And so the structure of your tRNA looks, it's often drawn like this. You have a line here, it comes up, makes a little bulb, boo, 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 boo. You have another one over here, you have a little spur over here, it comes down like that. So this is your tRNA. In your book, you have this diagram here, and it shows a bunch of different codons, boo, 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 boo. But it has an arrow pointing here and says that this is the mRNA strand. And it can be a little deceiving because you might be tempted to think that this part right here is, but that's not correct. I don't know why that happened in the book, but your mRNA is this guy right here. This strand here is your mRNA. You have a couple loops. They aren't super important for us to focus on right now. The two important ends are going to be this end right here and this segment over here. And on the left-hand side, we have what's called an anticodon. Now, if you think about the word anticodon, you have anti, which means not or the opposite of something, and then you have codon. So we all know our complementary base pairings. We have T goes to A, C goes to G, and then mRNA. Instead of T, we have U going to A. So because we're talking about RNA, we can forget about this guy, but these are our base pairings here. And so your anticodon, we know a codon is 3 nucleotides. So let's say we have C, G, A as our codon. Our anticodon will be the opposite of those. So for C, we'll get a G instead. For G, we'll get a C instead. And for an A, we'll have a U. So let's say our tRNA here, we have that. If we have C, G, A, our anticodon on our tRNA is going to have our anticodon. So it's going to be G, C, and U. And what that does is it allows it to attach and to mesh and to translate what we're seeing in our mRNA to a specific amino acid, which will be attached over here. Let's make sure there's nothing in the textbook that they want you specifically to know. So we talk a little bit about activator enzymes, and what an activator enzyme does is it attaches a polypeptide, an amino acid, there, to your tRNA. Now, another way to draw a tRNA, which is a little more accurate, maybe, it shows more of a 3D view, is it kind of looks like a goofy shape like that, where you have your amino acid up here, and then your anticodon is going to be down here in this segment here. And so what your activator enzyme does is your activator enzyme is what is used to take a little amino acid that's out there and match it to the proper tRNA that has the proper anticodon that will code for it. And so if we look at our big old chart from the other week that we use to decode our codons, you'll notice that while there are a bunch of entries in there, we have the redundancy. And so everything doesn't have, excuse me, every code doesn't have its own unique amino acid. There are multiple of them, and that leaves us with 20 total amino acids. And each activator enzyme is responsible for attaching one specific type of amino acid to the proper tRNA that's associated with it. So we have 20 different activator enzymes because we have 20 different amino acids. Well, now we know a little bit about tRNA and what it does. Now let's talk about how polypeptide chains are actually built using tRNA. A couple terms too that aren't in our book but are, maybe helpful to know is what we call charged. So once your activated enzyme attaches your amino acid to your tRNA, we call it charged. If it doesn't have your amino acid anymore, we call that uncharged. Now your mRNA is a little strand, and your ribosomes, what happened is it's built of two parts, right? We have the large unit and the smaller unit, and there are two places for your tRNA to sit in them. So you have one site here, and then you have another site here. And they each are referred to with a letter. The one to the left is going to be your P site, and then the one to the right is your A site. Now your ribosomes attach and start moving when they see a start codon. And our start codon is what? It encodes for MET, that's the amino acid, and the codon is going to be AUG. Excuse me. So we have our little well, let's try that again. So we have our little tRNA here, and we have our little RNA here. So our RNA is going to have, our mRNA is going to have AUG on there. And our tRNA here is going to have its anticodon. So the opposite of A is going to be U, the opposite of U is going to be A, the opposite of G is going to be C. So it's going to have U, A, and C on our anticodon end there. And then what is it going to have on our amino acid end? Well, we know that the start codon, AUG, gives us methionine, and so that is going to go on this end of our little tRNA. So now our tRNA, it goes and it matches up here. We'll get rid of this. A is kind of in the way. You'll see your U matches with your A matches with your U, your C matches with your G, and we have methionine on the top there. Now, that's all fine and well. It starts off there, but what happens next? Well, it's in the A site, but there's nothing else to bind it to yet. Let's think of another codon. So let's say the next thing we have on our mRNA is say CCU. So if we have CCU on our mRNA, our anticodon for that is going to be UUC. And so let's say we have another little tRNA, and it's got our anticodon, it's got our U, and C, and that is going to be proline. So you know, a little red one there. So what's going to happen now is instead of, well, I'll say it like this. Your mRNA here is linked to your tRNA through these bonds right here. And so it's not really moving anywhere yet. But what does happen now is our ribosome unit shifts. So this goes over here, and this guy goes over here, and this is our P site here, this is our A site here, and now our first unit, our first tRNA, has moved from the A site to the P site, which gives space now for our second tRNA to come in and match in this A site. And same thing here, we see that we have our C and our U, they match, they form bonds, our U and our C and our U again, which is pretty cool. Now we have both sites filled, and what happens now? Well, there's a bunch of different enzymes, and what they do is they start to form a bond between these two amino acids here. So those two become linked, and then let's draw ourselves another codon. Let's go with let's go with maybe A. So now, if we have A, our anticodon is going to be U. So we'll draw our little tRNA, little and our UUU is going to be lysosine, I think is how you pronounce it. But that's going to be that guy. And it's going to have UUU. Oops. This should be a here and a here. Sorry about that. That's my bad. Okay, so we have another tRNA and another codon that's ready to go. And what's going to happen now is, just like before, our ribosome is going to move over one. It's going to boop. That's going to move over there. This is going to move over here. Now, let's move this A out of the way. What happens now is, because these two here, they have that bond in them, the bond now breaks between your tRNA and the amino acid. So this guy ends up floating off into space with this little anticodon, where an activated enzyme can go and attach another met to this. But for now, it is in its uncharged state. So we'll just set that off to the side there. And what we have here now is we have these two amino acids that have started to form a polypeptide chain. which is a chain of multiple amino acids. And we have 2 now, so that's multiple. And now we have this chain, well, this codon now of AAA. So what happens next? The same thing as before. This tRNA is floating around, it goes, and then it forms little hydrogen bonds that match this guy and this guy and this guy to hold it there in the ribosome. And then we form another bond here. A bunch of enzymes do that. And then we're pretty much rinsing and repeating. Let's say our next codon is GU. G, that'll give us C, A, C, and we'll make a little tRNA for that guy. These are hard to draw. Okay, so our anticodon is going to be CAC on this end, and this is going to get a different amino acid, which is valine. And again, here we're going to see this guy here. Whoop. We're going to have this guy here move over to the P site. This tRNA here in the P site is going to break the bond between itself and the amino acid, and it's going to float off uncharged to get recharged somewhere else. And we're going to have this chain now of 3 amino acids, and this guy here is going to come in, and as the ribosome moves on to the right, this guy is going to get matched up with its anticodon. So we have our codon on the mRNA there. Again, our ribosome is going to move to the right. And this little tRNA is going to have to fly off, completely uncharged. But now we have this chain of 3 amino acids now. And it's about to be 4, because this guy here is going to come in and it's going to form a bond between those. But let's say instead of having GUG there, what if we had one of our stop codons? Anyone remember what those were? Well, let's find one of them. There are 3 of them. And for our example here, we will use UAA. I dropped the pen. 1 second. So we'll use UAA as our stop codon. UAA. Now, what's interesting is that none of the stop codons are UAA, are UAG, or are UGA. None of them have tRNA assigned to them with a amino acid, because there is no amino acid. These are stops. If we look at our chart, there's nothing, there's no little amino acid attached to these guys. So what happens is we have something called I believe it's called stop factor or a halt factor, which is something that it looks like a tRNA, and so we'll do, And what it does is it fits in here, right in here, and then the ribosome keeps on moving. So instead of bonds being formed up here, there are no bonds to be formed because there's no amino acid. Our little ribosome is going to move over. And again, our tRNA for this guy is going to go float off uncharged. And what that is going to do is it's going to leave us with this beautiful little polypeptide chain. And this guy will just get spit out as the ribosome moves to the right. So hopefully that all made sense. It's a pretty involved process, but it's nothing too complicated if you take it step by step. Big things to note. Make sure you know what an anticodon is and how to make one from a codon. You just find your codon, your three nucleotides, and then what you'll do is you'll find the complementary base pair, and that becomes your anticodon. Do note that things start in the A site and then move to the P site as the ribosome moves. The mRNA doesn't move, the ribosome moves across it. And while we just looked at this with one example of one ribosome, we do have polysomes, which is what happens most of the time. You'll have multiple ribosomes attached, and they'll start moving, and they'll start growing. And once one ribosome is out of the way of your start codon here, another one will take its place, and you'll have a big chain of them making the same protein and polypeptide chain. Make sure you're familiar with these three different purposes or functions of RNA that we've looked at so far. We have our mRNA, our rRNA, and our tRNA. mRNA is our messenger, rRNA is used to make ribosomes, and our tRNA is used to transfer and translate amino acids to our ribosomes. Alrighty, now an important aspect, or something that's just crazy, is how all this works. The more I think about it, the more I learn about it, study it, is like, how in the world do these tiny little things just move? How do they attach? How do they bind? How do they get from one place to another? And so it's completely normal if you have a bunch of different questions about this content. One of the big questions I had was, how does your tRNA up here, that's just in the cytoplasm, how does it get to the ribosome? Does it have like a little propeller or something on the back that moves it? Or I don't know, how does it get from one place to another? And it's interesting because there are a bunch of different ways. It uses diffusion and concentration gradients to move around, and it just ends up being in the right place where it needs to be at the right time for the ribosome to keep on moving and keep on building. Yeah, you can get a ton of interesting questions. If you have any, do drop them in the Google Classroom, and we'll try to get those answered for you. Let me just make sure we aren't missing anything that you guys are going to need from the textbook. Talked about most of our vocab, our A site, which is where the tRNA starts, and it's there with the amino acid. It's named after amino acid. The P site is our second site, which is where the polypeptide chain grows, which is why it's called the P site. A polysome is just what we talked about up here. It's a bunch of different ribosomes attached to the mRNA strand. Our transfer RNA are strands of RNA that transfer and move amino acids to ribosomes so that they can be coded. Now, one thing to note, which is pretty interesting, that I believe you'll have it's either a question in one of the quizzes or the worksheets. It says that tRNA is partially blank. So it's like tRNA is partially, and then I'll have a blank there. I think that's how it's worded, something similar. But if you look here, you'll notice that when we have our DNA, you always have two strands, right? And then our mRNA, it's just a single strand. Our tRNA, however, some places, like in here, and in here, and in here, and in here, there are two strands. But places out in here, and over here, and here, it's only one strand. And over here, that's only one strand. And so tRNA is partially double-stranded. It's not fully double-stranded, it's partially double-stranded. For the lab this week that we missed, you won't have to do a lab report or anything. It was pretty much just observation of slides. And so we can see if we can try to blitz through some of that next time we meet, just so you can take a look at what they're talking about here with protein translation in an organism. Make sure you're familiar still with our central dogma and what that means and what it's trying to show there, how our DNA gets transferred, gets transcribed, copied over to mRNA in the nucleus of the cell, and then it goes out into the cytoplasm where it's translated into a protein. And how that works, we now know. It uses ribosomes and tRNA and that mRNA strand as a template to build nice long polypeptide chains which form proteins. Yeah, I think that should cover it. It's really interesting to look at what tRNA actually looks like. Let's see if we can find a nice. So this is our common little diagram of it, but if you look at it, this is what its structure looks like. You can see some places over here in the yellow bit on top, it's single-stranded, and then it's double-stranded in other places. But this is what it actually looks like. Much more so than this diagram on the left here that we have. Here's another great image of what it looks like in real space and all your bases and stuff. Yeah, pretty crazy that's what is moving all your amino acids around to make proteins. Your ribosomes are also pretty crazy. Yeah, this is what they look like. So that's your large subunit and your small subunit. You can see it's just a craze of just rRNA mixed with proteins, which is pretty wild. It's just it looks like a big jumble, a big mess of nothing, but that's the unit that goes through and makes your proteins. It's got sites for your tRNA, and it builds your little polypeptide chains one by one. Take a look at a polysome real quick. Yep. Here you can see you have a bunch of them on your mRNA, and they start from one end and move to the next. All of them at the same time, encoding that. This is pretty cool. Um, yeah, that should do it for today. Hope you did well on the quiz. Hope you understood the material this week. If you have any questions, definitely drop them in the Google Classroom, which I finally have access to as of yesterday. And yeah, have a good break. Enjoy it. Don't forget to study your bio because we'll have a nice, fun quiz when you guys get back. Alrighty, good luck.